Thank you. Good morning, everyone. So I'll be talking about a project which is based on the University of Glasgow, but in collaboration with Nelson Mandela African Institution of Science and Technology in Tanzania. So I'm Divine Ekwem, and I'm based at the University of Glasgow. And today I'll be talking about how we've used information on livestock movement to explain foot-and-mouth disease transmission and spread, and how we can use that information to improve control of endemic FMD. Right, the problem hasn't gone away. FMD is still very there in sub-Saharan Africa. This picture was taken just several weeks ago in an outbreak we are currently tracing in the field. And what we have noticed is waves of these outbreaks, and with each wave, there is a new dimension of poverty, because these people in these communities, they totally depend on livestock for their livelihood. And what we've noticed is significant loss in milk production and also drought power, because they have to use this animal to cultivate crops. And of course, if you take this type of animal, the picture you're looking there in the market, you're very, very difficult to get any very decent price for it. So, following up to what Kim have said, in East Africa at the moment, we know that cattle, or well, I would say livestock generally, are more important, than wildlife, as opposed to the southern part of Africa, where wildlife still play a very important contribution to the FMD epidemiology. So when you talk about livestock factors, movement comes into the first thing you would think about. Movement have been implicated in so many outbreaks in the world, and it's not different in endemic setting. In East Africa, for example, livestock are kept in a very traditional system. You have the agro-pastoral list, which are located in the western part of the national park. And the agro-pastoral, they plant crops, and they also keep animal. But crops is very primary, and animals are just kept for secondary, just to subsidize income. While the pastoralists, which are located in the eastern part of the national park, they are those guys that keep animal and they keep plant very little crops. So there are different characteristics of movement, and I'll be talking to you through my slide on how these different movement characteristics will lead to foot-and-mouth disease transmission and spread. So in the short distance movement, you will see mostly it's when they go to those shared resource areas, while in the long distance movements, it's mainly associated with markets. So how do we collect the movement data? We have 3 field activities which are currently going in the field. So the first is the community pasturability mapping, which I'll be talking to you in detail, because that's the data I'm presenting today. However, we also have the GPS coloring, which is feeding onto the information generated from the participatory mapping. As you can see there, the green area is a grazing area, and then the red dots are the various areas where we have GPS colors. So we've colored animal at various distance to each grazing area, and we're looking at frequency of contact and also frequency of overlap. So we want to be able to relate that into our FMD transmission model. And of course, you can't talk about movement without involving the market. So we are also collecting movement permits to have a complete picture on how these animals are moving. Right, so talking about the participatory mapping, it's actually done at village level, because in this area, village is more or less like an epidemiological unit. Within a village, animals are likely to mix very quickly with each other. So when we go to a particular village, we present them with a map of that village, and then we recruit a focus group, which is those individuals that actually keep the animal, right? The large head keeper, the livestock traders, and then we ask them structured questions in terms of where they move the animal across seasons, from January to December. So they identify all these areas on a map, and then after the whole process, we take the village leader and said, can you confirm what these areas are? And then he showed it to us, and then we validate them with our GPS. So at the end of the whole process, which takes about 4 hours, this is what we get. So you have a map like this. Sorry, okay, yeah. So you can see that it was, you're right, there's a rice paddy field. So in the wet season, that is used to plant rice. But in the dry season, when they've harvested the rice, then it becomes a grazing area. While the other one there is a permanent grazing area. No one plant crops there. So all season you will find grass in that particular area. So this is the type of maps we have, and after the whole process, we digitize this map in GIS and then link them up with the qualitative information, and then we start drawing network. But before I go into the network, I will just summarize what local movement looks like in East Africa. There are 4 categories. You have the watering points and the grazing areas. These involve the daily movements and very high frequency of direct contact, indirect contact, and overlaps. However, the dipping points are also very important. In typical Serengeti district, which is an agro-pastoral community, you have got 555,000 cattle, and which are spread across 93 villages. There are only 10 functional dipping points. Dipping happens twice a month. So for each dipping day, you've got about 2,000 to 3,000 cattle that aggregate in one point from 6 o'clock in the morning to 6 o'clock in the evening, which of course become very integral in pathogen transmission. The sorting points are also important. However, they also buy commercial sort, so sometimes you see very few less aggregation in those areas. This is what local mixing look like. The red dot is the village, the blue is the shore resource area. So you can see how they are mixing there. Why the gray areas is just those connectivity. So sometimes you have some resource areas that are just shared maybe by one village, while you see others there is more aggregation in them. We kind of collapse the network and then redraw it just to look at inter-village connectivity. You can see the two red dots here, those ones towards the bottom. Those are two villages and they are connected at one point, which become a hotspot for disease transmission. And that is what transpires throughout the network. However, that differs across season. In the dry season where there is reduced water availability and open pasture is drying up, you see there is more aggregation because livestock need to travel longer distance and every village is connected to another village, every resource area is fully utilized. Why in the wet season? Because there is pasture availability, water is accessible from multiple sources. You don't see that mixing. There are some villages that are self-sustaining. They have got water, they have got pasture, they don't need to move. However, we now look at average distance across these particular communities and also how they differ from January to December. So what we notice is in the wet season, for example, within 5 kilometer radius, all villages are likely to connect with each other. But in the dry season, as the pasture level become low, water level become low, they are traveling longer distance, interacting with more people, and then the distance of connected villages widens. So that gives you an indication of what will happen if there is an outbreak in the wet season. Within 5 kilometer radius, everyone is likely to be connected with each other, and then disease is likely to transmit within that community. The transition period are the June and October, which is from wet to dry transition, of course, and then from dry to wet. Right, so we use community dictation algorithm. Sorry, I don't know if the picture contrasts there very well, but we use community dictation algorithm to look at the different in the connecting structure of the pastoral and the agro-pastoral communities. So what you can see here is each polygon so each polygon here is a community of villages that are closely connected with each other. So what happens is the connectivity within them is very strong. Like in the agro-pastoral community, a modularity score of 0.6 is telling you that communities in the agro-pastoral settings, they are tightly connected with each other. And the reason is because they plant crops and they have few areas of grazing. So those few areas where they have for grazing, there is high livestock density and high livestock aggregation. While in the pastoral communities with a low modularity score, you see that the villages, the communities are highly connected among themselves, but connectivity within community is actually much less. So we now use this information to actually investigate an outbreak. These are just the basic protocols which we follow. And then coming again back to the Serengeti district, which is agro-pastoral community. It's located very close to the border with Kenya. And what we have noticed, we have active surveillance going on in these communities. And for the past 20 months, we haven't noticed any outbreak. None was reported so far. And then it eventually happened. So I was lucky to be in the field when it happened. In that village there, we were told there is an outbreak. So we arrived. For almost 20 months, there hasn't been any. Even the villagers themselves, they thought maybe FMD had gone away. But it's not going away without any solution to it. So there was there, and then we started collecting sample. 3 days later, we got another call that there's an outbreak in the next village. So we got as well. And by the time we complete our history taking, we realized that the timeline of possible exposure was almost maybe within 1 or 2. So it's possible that simultaneously these two villages might have become infected. As we're doing the contact tracing to retrospectively trace back, where contact history might have come up, change in behavior that might have led to exposure, of course, it keeps spreading to the next village. So we keep tracking the outbreak until we eventually got at this point. So the red little dot there you're saying is when we complete the investigation and we realize with detailed contact history that that was actually the village that the first outbreak happened. So we got into there and we spoke to the gentleman and he said, yes, for the past 20 months, we haven't noticed any outbreak. And I think I was the very first person that had it in this community. And I had done something which I don't normally do. I have traveled 15 kilometers to buy animal in another district. Now, in these communities, when they acquire animal which they introduce into their heads, which is very rare because they believe in natural birth to improve their head size, but 30% of them normally buy animals from local markets. And when they buy from those local markets, it's mainly market within 5-kilometer radius, which you've already mixed anyway in the resource area. So nothing is new. But when you involve in this very risky activity of traveling such a long distance to buy an animal, you are likely to cause this kind of situation. So we now overlay our network to this investigation and we realize that there's something very peculiar with these 3 villages. The very first one that the outbreak happened, and then these other 2 where we first noticed outbreak, which was very severe, number of villages, almost every household was infected. And we realized they share this grazing area. And also the next one that is adjacent, they actually have a water hole which they share. So we think this is what is really happening. Once you are getting into this very risky behavior of a long distance movement, acquiring animal from a very far district, introducing to your head, it gets spread into a grazing area, and then you start having this local spread incident. So we overlay that with our network, and you can see within that community, that is where that virus was spreading before it could escape into the next community. So in summary, we have identified areas that are very critical, which we can focus our control strategies on, which I'll be talking to you in a moment. And we'll also validate our network and realize that for an outbreak to be reintroduced into an area that you haven't had outbreak for such a long time, there's likely to be 2 activities: acquiring an animal from a very distant market and also the sharing of communal resource areas. Of course, we can control movement of animals in these areas. It's very difficult. However, vaccination remains the ultimate control goal, but we are still struggling because we don't have any that we are using in East Africa at the moment. So, looking at the network there, you can see the big red dots. They show the villages with high centrality measures, which are just those villages that are very important in maintaining the bridge across the network. Several studies have already demonstrated that if you focused strategy on those villages, maybe you vaccinate 10%, you're likely to reduce an outbreak by almost 80%. So we think this will be very effective in a typical endemic setting where resources are very low. So you're talking about 555,000 cattle. How many vaccines are you going to use to vaccinate them? But if you could focus your strategy on this information we have now developed through this network analysis, then that will really help in paving a pathway for FMD control in endemic area. And of course, we are shipping this shipment to reference laboratory to be able to confirm the virus and potential vaccine match. And of course, we've been very interested to see what is really happening during those long, extensive periods where there are no outbreaks. For 20 months, there are no outbreaks. So what is going on? So we hope to use serological data because we have GPS heads. The GPS study I showed you, we actually have heads with GPS, which we are sampling every 3 months. So the plan is to be able to see what is really going on in those animals when there are no outbreak. And then immediately when there's an outbreak, how does that change the serological picture? And of course, we have models that can actually infer history of outbreak. So we also plan to complete our GPS analysis and also integrate market data fully into the network. So I'd like to thank people from the United Kingdom and those in Italy, and also special thanks to MSD that are supporting our field activities. We are very grateful, and of course the Bill and Melinda Gates Foundation that is sponsoring the project, and the African Bioservices as well that is providing support with the GPS coloring work. And of course, thank you very much.
For one question for Divine.